microbe from replicating. We can block RNA polymerase, so it prevents the formation of messenger RNA. You don't get protein synthesis occurring. Interfere with metabolic pathways. So if you interfere with glycolysis or the Krebs cycle or the electron transport chain or folate synthesis, which is what your book talks about, if you interfere with any one of those metabolic pathways, you can again kill or stop the spread of the microbe. Damage the cell membrane integrity. Oh, I forgot to write the, this is example of a metabolic pathway, and if you interfere with that, you can interfere with microbe growth. And then finally, what can you do to the membrane? Can you do something that punches holes in the membrane so that the cells lyse? What we need to recognize is that drugs are going to have different effects depending on the microbes that we're targeting here. Penicillin is going to be very effective against gram-positive microbes with the thick peptidoglycan wall. Less effective against gram-negative microbes because they've got that outer cell membrane, that outer membrane that kind of interferes with penicillin getting to the peptidoglycan. Those that target cell membrane integrity, they're going to be more effective against gram-negative cells because they have that outer cell membrane that can be targeted. Less effective against these gram-positive cells.
What was more effective?
Say that again?
The damage to the cell membrane is more effective on gram-negative.
Yeah, because they have that outer membrane that can be targeted.
But which one does? Which cell membrane integrity?
Cell membrane integrity.
Which one was the one that was good against gram-positive?
The cell wall, when you're using penicillin, because it's accessible to it. Keep in mind, and this is always going to be the case, you've got to have contact for any of these to work. They have to come in contact with the microbe. The chemicals have to come in contact with the microbe to do any of the damage that we're talking about. Your book also talks about specifically tuberculosis, Mycobacterium tuberculosis, because it's a unique microbe in that it has a waxy molecule in its cell wall, a mycolic acid, that makes it resistant to a lot of drugs. And we're actually seeing more resistant strains of M. tuberculosis as we go on. There are 5 main medications that are used called the first-line drugs to treat tuberculosis, and they're used in combination. And this is because of the concern for resistance, the developing of resistance. If you're using one medication, one of a few microbes might mutate, or you might get a few that are resistant to that one medication. But if you're using a second medication, that probably is going to wipe out those that are resistant to the first one. And if you're resistant to the second one, you're probably not resistant to the first one. So in a combination, you get rid of cells that might have some resistance to one drug or the other. A couple of things to talk about with determining the effectiveness of drugs. We've used one of these methods in class already in lab. So if you wanted to determine the susceptibility, we can look for what is known as the maximum inhibition or inhibitory concentration, MIC. How do we do this? We have to set up what is called a serial dilution. This is something that we're going to do in lab here real soon. But basically, you set up yourself a number of tubes, and depending on the series you want, and then you decrease your chemical concentration, starting with a control that has nothing in it. You could start with something that's 100%, down to 50%, down to 25%, down to 12.5, 6.25. You could take this all the way down to whatever percentage you wanted, or you could use a concentration. You could start out with, say, 10 milligrams per liter, 9 milligrams per liter, 8 milligrams per liter. But you'll have a series of concentrations, and then you add the microbe, okay? And so now we're exposing the microbe to these concentrations, and you determine at what point do you see growth. And when you see growth in that tube, that'll tell you the next tube over, where there is no growth, will give you your minimum inhibitory concentration. And so this allows us to determine how much chemical is needed to kill this microbe. It allows you to determine a dosage. This is a quantitative method for determining the susceptibility. In lab, we did a qualitative method as well, called the Kirby-Bauer method. If you remember, in this case here, we gave ourselves a bacterial lawn, and then we put discs that had been soaked in and disinfected. You could do the same thing with an antimicrobial, and then we looked for zones of inhibition around those discs. In this case here, we're doing a qualitative assessment. We're not really quantifying how much chemical is needed to kill. We're just determining which of these kills best. Now, we could, if you wanted to, set up solutions such that you've got 100% solution, a 50% solution, a 25% solution, and you could soak these discs in those different solutions. And so you can quantitate it that way. But for the most part, we're talking about qualitative analysis. Brings us to drug resistance. So resistance can happen fairly rapidly. Think about how this can occur. So you've got several microbes, and you're being given an antibiotic to treat these microbes. And here's the deal. Most of these are sensitive, except this one here. This one's resistant. Okay, so your antimicrobial wipes these guys out. They're dead. But either this one is able to survive past the treatment process, or you stop taking your medication early. So now this is the one that survives, and so when it reproduces, the new population is that of resistant microbes. Like so. What you have to recognize is that when we talk about a population of cells, population of organisms, anything like that, if you're talking about a particular characteristic, so in this case, if we were talking about sensitivity to a particular chemical, there is always going to be a range of sensitivity within that population. Just like our population, there's a range of sensitivity to certain viruses or bacteria. Same is true here. And that range is almost always in the shape of a bell curve, where you're going to have on this side those that are extremely sensitive, and so these guys get wiped out from almost the first exposure to the chemical. And then on this side, you're going to have those that are extremely resistant. And then you have all those that are in the middle, and generally are going to get wiped out by the chemical. But in almost every case, every population has this kind of distribution, with some that are always going to be more resistant and those that are going to be more sensitive. Okay? And so when you allow for that resistant strain to develop and evolve, then you can get cases where the microbes now are no longer responding to the treatments that we're giving them. Such is the case with MRSA, Staphylococcus aureus. 50 years ago, only 3% of the cells showed any kind of resistance to penicillin, any kind of resistance to methicillin. Today, over 95% of the strains are resistant to penicillin. That's a development of resistance just over the last 50 years or so. Significant. And we're running into a problem now, because now we have methicillin-resistant Staphylococcus aureus that is spreading through the prisons and the schools and stuff. And the drug that we've been using, vancomycin, they're now starting to develop resistance to that. So you're going to be hearing about not just MRSA, but VRSA as well, vancomycin-resistant Staphylococcus aureus. So let's talk about the mechanisms that a microbe would use to develop these resistances. So normally, if you think about the action of an antimicrobial, what you're talking about is that you've got the drug, and the drug is going to diffuse into the cell, be taken up by the cell, make its way into the cell, and there's usually a target compound. The drug will bind to that target compound, and essentially this compound now has been eliminated. If it's necessary for a metabolic pathway, a protein synthesis, DNA synthesis, something like that, it's going to interfere with that pathway. So this is what we would expect from a non-resistant cell. But when we look at a resistant cell, here's what we see. In some cases, the chemical doesn't get taken up. The bacterium is able to reflect it, and so there is no uptake occurring. How does that happen? Well, maybe it's changed one of the surface proteins that the compound bound to, or maybe the channel that it used to flow through has changed, so it doesn't fit anymore. Something the microbe has done has changed, and so now the chemical can't get into the cell. You could eliminate it immediately. So as the chemical comes in, the microbe quickly flushes it out. We talked about this very early in the semester, a process called efflux. So as the chemicals diffuse into the cells, they are pumped out before they have a chance to interact with the target molecule. Sometimes the chemical will diffuse into the cell, but now the cell has changed the target molecule, and the chemical can't bind to it. This can happen by way of mutation, different plasmid proteins being produced. We could inactivate the drug. Some bacteria have the ability, the drug moves into the cell, they bind to that chemical such now that it cannot carry out its activity. So our target molecule's over here, you can't bind to it now because it's bound by some other compound. So a number of ways that the cell can evolve to develop a resistance to the chemical that we might be using. These resistances, they come about through spontaneous mutation. I mentioned this already. This occurs at a fairly low rate, but still does occur. One of the main ways that you can get resistance to develop is through plasmid transfer. We talked about this before. Gene transfer via plasmids could give